I am Chris Holzapfel. I'm the Holzapfel part of the Bradshaw-Holzapfel Laboratory of Evolutionary Genetics. I have spent the bulk of my research career at the University of Oregon, having come here directly from my postdoctoral work at Harvard University, bypassing a tenure-track position at the University of Michigan, which I will admit was not the easiest decision I've ever made, but in hindsight it was a really good one. Bill Bradshaw and I have been able to develop a novel system together in which we have been able to answer transformative questions for more than 30 years. Today we are going to talk about the work that is ongoing in our research right now. We have 3 arms of research that we are focusing on. One is on the eradication of blood-borne disease. The other is on biological timing mechanisms, that is the integration between the daily timer that actually allows you to get up in the morning and the seasonal timer that tells us about how the world looks around us. And finally, climate change and the observations that we made relating to climate change penetrating to the level of the gene and how that changed the understanding of that phenomenon. So what is this novel system with which we are working? It is the pitcher plant Wyomia smithii system. Wyomia smithii is a little black mosquito, kind of nondescript, small, that lives only in the water-filled leaves of the purple pitcher plant, Sarracenia purpurea. This is a carnivorous plant. I'm actually surrounded by them here. And this mosquito spends its entire pre-adult development and life in the leaves that normally eat insects for breakfast, lunch, and dinner. So for 2 young scientists starting out, it seemed like this had to be a system that was going to tell us many interesting things. Wyomia smithii spends pretty much its whole pre-adult development in these leaves and then emerges, flies around, makes whoopee, lays eggs, and the whole cycle begins again. When we started out, we obviously knew very little about the Wyomia system. We now know more than you're going to even want to know, but I'll tell you a few things that are important to the understanding of what we are doing. 1, it occurs over a huge geographical gradient. It occurs all the way from North Florida up into northern Canada. At the same time, it also extends from sea level into the highlands of North Carolina. So it has a huge latitudinal range and a huge altitudinal range. For geneticists, this is very useful. We know a great deal about its historical geology, and very importantly, we know the relationships between the more than 100 populations that we have over this range, all the way down to the nucleotide. So therefore, we are able to ask and answer comparative questions at the level of the genome. Currently, there are many, many labs, probably thousands worldwide, that are focusing on individual diseases and how to mitigate the effects of blood-borne disease. Every one of those labs has one thing in common, and that is that they assume that a bite is going to occur. So we took our system of Wyomia smithii that lives in this pitcher plant and said, okay, so what can this system tell us that's different? Bill and I have known for 20 years easily that Wyomia smithii is the only mosquito that has been described out of the thousands of mosquitoes that are known that bites in one part of its range and is an obligate non-biter in the rest of its range. So we are able to actually extend the discussion from the level of biting to what happens if there is no bite. If there is no bite, there's no blood-borne disease of any kind that is transmitted, period. So no bite equals no disease transmission, period. The secret that we're looking for is held somewhere in the middle. The genes that are involved in the biting phenotype are somewhere on the transition from biting in the south to obligate non-biting in the north. What are these genes? So we have used the most modern genetic and genomic tools and bioinformatic tools to bring the number of genes that we have from many down to about 300 genes that are involved in the biting phenotype. Once we get the number of genes to about 100, then we are going to work on the verification of those genes using a technique called RNAi or RNA interference. And basically in gene verification, the genes that are intimately involved in the biting phenotype are kept, and those that are supplementary to the biting phenotype or less deeply involved in the mechanism are not kept. We already have several pharmaceutical companies that are interested in developing non-toxic inhibitors that will change effectively biting mosquitoes into non-biting mosquitoes, and that will be our next step. And if those pharmaceutical companies are able to succeed in this process, we will have changed the direction of medicine for thousands of people worldwide. Before I turn this over to Bill, I do want to say one quick thing, and that is that if you're actually working right on the cutting edge of science and are asking a really good question, a lot of other labs are going to be asking that same question. Thus, what we always do at meetings and the like, we find out how are other labs approaching this question, and then we do something different. We take our mosquito system and say, okay, YMI Smithii, what can you do to tell me what is different than what everybody else is doing? If you aren't asking something in a different way, it's very unlikely that you are coming up with a new solution to a problem that will truly transform an area and move discovery forward.
I'm Bill Bradshaw, and like Chris, I came to Oregon some 30 plus years ago from a postdoc at Harvard. My topic to begin with is going to be about biological timing mechanisms. So what are biological timing mechanisms? It's all about when to do things. First of all, we, like the rest of the world around us, have a daily circadian clock that tells us when to do things in general during the day. There is also the seasonal photoperiodic timer that assesses the length of day or night and essentially tells organisms what time of year it is. Both of these physiological mechanisms are internal to the organism, and they both share the property of anticipation. They enable the organism to prepare physiologically for future events, both on a daily basis and on a seasonal basis. You can think, well, summers are warmer than winters, why not just use temperature? But temperature is actually a relatively crude cue. Temperature varies from week to week, month to month, and is a very inexact cue of future seasonal change. On the other hand, the day length where you're standing right now today was the same 10,000 years ago as it is today and will be 10,000 years into the future. In sum, the length of day is the most reliable cue for future seasonal change through evolutionary time. Historically, the daily circadian clock and the seasonal photoperiodic timer have been considered as a single integral mechanism. That is, the daily circadian clock forms the necessary mechanistic basis for the seasonal photoperiodic timer. Other schools of thought have proposed that they're not connected at all. And indeed, the theoretical and experimental results from our lab showing that they have evolved independently over the climatic gradient of North America has really changed the way people in biological clocks and biological timekeeping have approached their research. We have proposed that a single or a few genes in the daily circadian clock can be giving a time cue, a time reference point to the seasonal photoperiodic timer. That is, one or a few genes in the circadian clock as they cycle, independently and incidentally of their role in the circadian clock, give a time reference point to a separate independent photoperiodic timer, which takes that time cue from the daily circadian clock. We do know that regardless of how it turns out, this is a very important distinction to make, because the health and well-being of human populations, the agricultural plants and animals upon which we depend, and conservation measures to preserve and conserve the world of nature about us as we know it are dependent upon these two timing mechanisms. The third aspect of our research that we are currently emphasizing concerns global warming. Few scientists would question the fact that the Earth is warming and is doing so at a rate that is faster than it has done in the previous 1,000 years. If global warming were having an impact on Waimea, how we would expect it to do so. And what we have found when we look back at our own data is that northern populations are becoming more southern-like in their response to day length. And this shift in response to day length breeds true in the lab. That is, that recent rapid climate change has penetrated to the level of the gene. This conclusion subsequently was confirmed, or has been confirmed, in insects, plants, birds, and mammals. This makes perfectly good sense because, unlike the physical world, the biological world is responding to the changing seasons. Growing seasons are now longer than they used to be. Winters are shorter and milder. That this is a seasonal adaptation by biological organisms. Yes, glaciers are melting and sea levels are rising, but animals are not glaciers, and the effects of global warming are on changing climates as far as the organisms are concerned. And it is this viewpoint that it is climate change and change in the lengths of the growing season which has changed the focus of the field when we are considering what is happening, actually happening to organisms in nature confronted with this rapid climate change. In sum, Chris and I are two independent scientists. We have complementary viewpoints and skills, and we have used the somewhat small, wimpy, otherwise nondescript, but totally magnificent mosquito, Waimea smithii, to resolve issues and move on to new and compelling questions. We have used throughout our career the most cutting-edge genetic and genomic tools available. As Chris said, unless you force yourself to confront what is new, you're unlikely to obtain results that are truly transformational and move discovery forward.